So, let's dive into a bit of a molecular mystery, shall we? It's the story of two molecules that are built from the exact same building block, but end up with completely different destinies. One is this vital source of energy we all need, and the other is one of the strongest materials in all of nature. How on earth is that possible? Well, let's find out.
And this all boils down to a really simple question, right? Think about it. Both a potato and a tree trunk are just packed with molecules made from glucose. So what's the deal? Why can one fuel your body for hours while the other is, well, it's completely indigestible? The answer is hiding in a tiny, tiny difference in their structure that changes absolutely everything.
All right, first up, let's talk about the hero of our potato, starch. The best way to think about starch is like nature's coiled rope. It's this brilliant way for plants to pack away massive amounts of energy into a really compact, accessible form. It's just sitting there, ready to be unwound whenever that energy is needed.
You know, what's kind of cool is that starch isn't just one single thing. It's actually a mix of two different forms. You've got amylose, which is this long, simple, straight-ish chain, and then you have amylopectin, which is a much bigger, way more branched-out structure. But here's the thing. At the end of the day, they're both just long chains of the same building block, glucose.
Okay, so here's the secret sauce. It's all about how those little glucose units are hooked together. In starch, they're joined by something called an alpha-1,4 glycosidic linkage. Now, this specific bond isn't perfectly straight. It actually forces a natural bend in the chain. So as you keep adding more and more glucose units, this little bend makes the entire molecule twist up into this beautiful spiral, or a helix shape.
And we can actually see the effect of this spiral in a super classic chemistry experiment, the iodine test. Turns out, that spiral shape of amylose is the perfect little cage to trap iodine molecules inside its coil. And when that happens, boom, you get this really distinct dark blue color, which is how you know starch is there. If there's no spiral, you get no color. Simple as that.
Okay, so now for the other side of the coin, cellulose. If starch is that coiled rope made for energy, cellulose is the polar opposite. You gotta think of it as nature's steel cable. It's not designed for storage at all. It's designed for incredible structural strength and rigidity.
And when I say incredible, I am not kidding. Cellulose is the single most abundant natural polymer on our entire planet. It's literally the stuff that gives trees their strength. It's the main component of wood and bark. It is everywhere, providing the backbone for basically the entire plant kingdom.
So what's the deal? How can it be so different if it's also made of glucose? Well, you guessed it. It all comes back to that linkage. Cellulose uses a beta-1,4 linkage, and this tiny change has a massive consequence. Instead of a gentle bend, it forces every other glucose unit to flip a full 180 degrees. This completely cancels out any curve, creating a perfectly straight, extended chain. And that perfectly straight shape, that is the secret to its incredible strength. Because the chains are flat and straight, they can line up perfectly next to each other, almost like you're stacking planks of wood. This lets them form all these powerful hydrogen bonds between the chains, basically locking them all together into this cross-linked, rigid, and immensely powerful fiber. All right, let's put it all together. This is the big showdown, the alpha-beta divide. You've got the alpha linkage creating a spiral for energy storage, and you have the beta linkage creating straight chains for pure strength. This single, tiny difference is the entire reason a potato is food and a tree is building material. So let's just lay it all out side by side. For starch, you've got the alpha-1,4 linkage. That creates a natural bend, which results in that spiral shape. For cellulose, you've got the beta-1,4 linkage. That causes a 180-degree flip, which creates a perfectly straight chain. That's it. The whole story is right there in that one little detail. And here it is, the big visual aha moment. On the left, you see that alpha linkage creating the coil of starch. It's open, it's accessible, and it's easy for our enzymes to break apart for energy. On the right, you see the beta linkage creating the flat, stackable chains of cellulose. So tightly bound together, our bodies literally can't break them down. One tiny flip, two completely different worlds. But wait, there's actually a third player in this game. Let's talk about glycogen. See, if starch is how plants store their energy, glycogen is how we animals do it. It's our body's own version of the coiled rope, but it's been optimized for a much, much more active lifestyle. Now, glycogen looks a whole lot like the amylopectin we saw in starch, but it's like starch on steroids. Its branching is far more frequent, way more dense.
And this structure is no accident. It's a brilliant adaptation for animals that need to get to their energy stores very, very quickly.
See, we store this stuff in our liver and muscles for a reason. All of those extra branches, which pop up every 8 to 12 glucose units or so, give our enzymes tons of places to start working. They can start breaking down the molecule from every single one of those branch tips at the same time. And that gives us that rapid-fire access to glucose for a sudden burst of energy, to run or fight.
So, to wrap up the main story here, let's just put them head-to-head one last time. You've got starch, the coiled rope. Its whole job is energy storage. That spiral shape is easy to unwind and break down. Then you have cellulose, the steel cables. It's built for structural support. Its straight, cross-linked shape is incredibly strong, rigid, and for us, pretty much impossible to digest.
And that really is the big takeaway, isn't it? The story of starch and cellulose is just this perfect example of how one tiny, almost insignificant change at the molecular level can create these monumental differences in the world we see and interact with every day. It really makes you wonder, what other simple molecular flips are out there, shaping the reality all around us?